Hi, my name's Adam Jacoby, and I'm here to talk to you guys about Legionnaire's disease. So, Legionellosis is the formal name for Legionnaire's disease. It's a type of atypical pneumonia, and it takes predominantly about 4 to 5% of all pneumonia cases each year. Now, there's another form of this called Pontiac fever, which is a less serious form of a Legionella infection, but it doesn't cause pneumonia. So the infectious agent is the Legionella bacteria. Now, Legionella pneumophila causes about 90% of all cases, and there's at least 35 different cervivars known, where the 1, 4, and 6 are the primary disease-causing strains. Other species members of Legionella can make up the remaining 10% of infections. Now, there's some main characteristics of the L. pneumophila bacterium. It's gram-negative, it's non-encapsulated, it's a mesophile, meaning it prefers temperature range of 25 to 37 degrees. It's rod-shaped and obligate aerobe. It's motile depending on the cycle. It has flagella, and when running tests, it's catalase positive, oxidase positive, nitrate reduction negative, and is a fermenter. And it has a pH ideal range of around 5.5 to 9.3. It's also 3 to 5 micrometers in length, and some serogroups are found to engage in biofilm formation. So here's a picture of the L. pneumophila bacteria. Just for reference, you can see the flagella and the rod-shaped pretty easily. And this was also the one that took place in the strain that was caused in the original identification of it in 1977. So speaking of that, it was not characterized as a disease until 1976. Outbreak occurred in Philadelphia at an American Legion convention, which is where it gets its name, Legionnaires' disease, because of the Legion. So there was 182 reported cases and 29 deaths. Now, in December 1976, Dr. Joseph McDade, who happened to work for the CDC, managed to isolate the bacterium causing the Philly outbreak and identified it as L. pneumophila. And the following year, the CDC gave Legionnaire's disease its official name. So after this, scientists needed to work backwards in order to see what previously undetermined cases could have been caused by Legionnaires. So they found that the 1957 breakout in Austin, Minnesota, in which it was a food company, that there was 78 cases of pneumonia. Then in 1965 at St. Elizabeth's Psychiatric Hospital, the bacteria was also found from later antigens that only respond to the L. pneumophila bacterium. And then in 1968 Pontiac, Michigan, where we get the name for the non-pneumonia form of a Legionella infection called Pontiac fever. And then in the early 70s, it ranged for a few years because it was tourists, the Rio Park Hotel in Costa Blanca. So, L. pneumophila has several different virulence factors. It has lipopolysaccharides, lipid A endotoxin, pili, flagella, secretes a bunch of different things, macrophage infectivity potentiator, or MIP genes, which just increase the infection ability of L. pneumophila, DOT-ICM type 4 secretion system, T4SS, which helps invade host and create signal molecules. Now, it's also an intracellular pathogen, and it's engulfed by alveolar macrophages, alters the endocytic pathway, and its main thing is that it inhibits fusion of phagolysosome, allowing the bacteria to multiply. So here's a diagram that shows the infection cycle, in which the bacteria enters the cell by first phagocytosis. Now, once the bacteria is phagotized, the bacteria occupies a unique phagosome. The phagosome does not follow an endosomal pathway, but instead utilizes its own novel pathway. This is where the type 4 secretion system shown in the image contributes to the infectious cycle. It serves in the infectious cycle of Legionella by encoding for mechanisms needed in the transfer of plasmid DNA. Now, The phagosome is able to evade lysosomes and endosomes because of a protein secreted by this type 4 secretion system and the markers it lacks on its outer membrane, so it's not recognized. While a phagosome avoids interactions with lysosomes and organelles, it fuses with the host cell's mitochondria and begins capturing endoplasmic reticulum exit vesicles. Now, around 4 to 6 hours after phagocytosis, the phagosome containing the L. Nemophila affiliates with the ER of the host cell. The ribosome-studded membranes have been shown to be ER from the host cell through the use of antisera to detect ER protein BIP. Up to 14 hours after the bacteria were phagocytized, the bacteria undergo replication. During this time, there's a phase called the post-exponential phase, in which the bacteria undergo changes in its phenotype and develop several very noticeable morphological changes, which occur mainly to the virulence-related genes. Sigma factor, FLA-A, is produced, leading to lysosome avoidance factors and secretions of adjuvant, leading to an acute inflammatory response. It is hypothesized that during the late stages of infection, the bacteria moving freely in the cytoplasm cause structural disruption in the cytoplasmic membrane of the host cell. The bacteria then exit through the pores to infect other cells in its ineffective form, and cell death through lysis occurs shortly after. The incubation period for the bacteria lasts about 2 to 10 days in the host, but typically it's only 5 to 6. Anyone can get the disease, but will most often affect the elderly, as 70% of cases are for patients 50 years of age or older. Now, early symptoms of the disease are often flu-like with muscle aches, exhaustion, headache, and dry cough. Part of the reason why it's so difficult to differentiate from other diseases. Now, this is followed by high fever with temperatures reaching 102 to 105 degrees Fahrenheit, chills, and sometimes diarrhea. When you take x-rays at this point, they'll very often reveal pneumonia. Now, the mortality rate is 40 to 80% in untreated immunosuppressed patients, but it reduces to 5 to 30% for those who aren't, depending on the treatment, how fast the disease is detected, and the severity of symptoms. In Europe, Australia, and the US, 10 to 15 cases are detected per million in the population. Now here's an image of lungs with Legionnaires where you can see the pneumonia right around here. Legionnaires is spread by inhalation of aerosolized contaminated water via the upper respiratory tract. Now there's no known cases of person-to-person contact, zoonotic, or animal-animal contact. And it's usually found in stagnant, warm freshwater areas, which are very often man-made, which these water sources can include, but they're not limited to, there's just anywhere that's stagnant. Lakes, rivers, ponds, water heaters, cooling towers, plumbing systems, spas, and pools, just to list a few. Now, there's no known vaccine available for LD, and cases with non-pneumonic form, Pontiac fever, you just treat the symptoms, there's no antibiotics used. And the treatment type depends on if the patient is immunocompromised, and it usually lasts 7 to 10 days. And the typical drugs used are tetracycline, quinolones, macrolides, which include azithromycin, erythromycin, clarithromycin. And the ones used for immunocompromised are usually these macrolides. Now, to prevent Legionnaire's disease, its regular decontamination of stagnant water areas should be done, since we know it comes from still warm water, fresh water, and that's why it's so important to provide clean water supplies. And the current research that is being done on Legionnaires is how Legionella's role in biofilm formation and cheaper, more efficient ways to identify the disease, and identifying it early, since it's so difficult to differentiate from other forms of pneumonia. Thank you for listening to this presentation.